Hello guys, today we are going to talk about the RBC, that is the red blood cells. Already we have seen that RBC forms about 45% of the blood. RBC is a bioconcave disc which is about 7 to 8 micrometer in diameter and the thickness is about 2.5 micrometer. Now let's see the structure of RBC. RBC is bounded by red cell membrane which has blood group antigens present on it. To this red cell membrane is attached an interconnection of protein network called the cytoskeleton of red cell. This cytoskeleton is made of spectrin, actin, band 2.1, band 3, band 4.1 and chirin and many other proteins. This gives the cell a bioconcave shape and a unique feature called deformability. Because of this unique feature, the red blood cell can pass through the tiny capillaries. Any abnormality or deficiency of these proteins can result in membrane abnormalities like spherocytosis. Now let's talk about the various normal parameters of RBC. The normal amount of RBC in human adult male is 4.5 to 5.5 million cells per cubic millimeter, and in females it is 3.8 to 4.8 million cells per cubic millimeter. The normal value of PCV, packed cell volume, is 40 to 50% in males and 36 to 46% in females. PCV is nothing but the measurement of blood made of cells. It is also known as the hematocrit. The normal value of hemoglobin in males is 13 to 17 grams and in females it is 12 to 15 grams. Further, I want to add that these values can slightly change depending on the various labs or the hospitals, but it's okay. So now let's discuss about the various red cell indices. These red cell indices are the average values for the red blood cells and is very useful in the diagnosis of anemias. So the first one is MCV, mean corpuscular volume. MCV means the total volume of red blood cells. It is expressed in femtoliters. The normal range is 80 to 100 femtoliter. If it's below 80, it's known as the microcytic, and if it's above 100, it's known as the macrocytic. So the next one is MCH, mean corpuscular hemoglobin. It is the amount of hemoglobin per red blood cell. It is mostly expressed in picogram. The normal range is 26 to 34 picogram. So the next one is MCHC, mean corpuscular hemoglobin concentration. So it indicates the concentration of hemoglobin in the red blood cell. It is mostly expressed in gram per deciliter. The normal range is 31 to 37 gram per deciliter. If it is below 31, it is known as the hypochromic red blood cell. If it's above 37, it is described as hyperchromic. Next one is red cell distribution with RDW. So RDW gives idea about the volume of red cell population. It mostly reflects the range of red cell size. So the normal range is 11 to 14%. The smallest size is mostly seen in fragmented red cells, and the largest size during the reticulocytosis or macrocytosis. So the next one is the reticulocyte count. So reticulocytes are the newly formed RBC released from the bone marrow. So it mostly takes 2 days to mature in the bone marrow, and these are in circulation for about 1 day. So it can be detected in the peripheral blood. So the normal range is 0.5 to 2%. So now, as we all know, that RBC is the bioconcave disc, but sometimes there is alteration in the RBC size and shape. So, but normally the RBC is called as normocytic, normochromic. Normocytic because the diameter is between the range of 7 to 8 microns, and normocytic when the central pallor is about 1/3. So therefore the normal RBC is known as the normocytic normochromic. Now let's see the various defects or the alterations in the size and shape of RBC. The first one is macrocytosis. So the RBC is called as a macrocytes when the size of the RBC is larger, that is the diameter is more than 8 microns and MCV is more than 100 femtoliters. It's mostly seen in vitamin B12 and folic acid deficiency. The next one is microcytosis. When the size of the RBC is small, that is the diameter is less than 7 microns and MCV is less than 80 femtoliters, it's known as the microcytosis. So it's mostly seen in iron deficiency, anemia and thalassemia. Now the next one is anisocytosis. Anisocytosis denotes the variation in the size of RBC. So in short, the RBC cells are of different sizes. So it is mostly seen in megaloblastic and hemolytic anemias. The next one is poikilocytosis. So poikilocytosis is the variation in the shape of the RBC. Like if you see in the smear, you can see a lot of teardrop cells, oval cells, or helmet cells, sickle shaped cells. So these sickle shaped cells are mostly seen in sickle cell anemia. Now the next one is spherocytosis. So in the smear, if you see, spherocytes are recognized as small, deeply stained cell. So central pallor is mostly not observed in these cells. So this is mostly seen in hereditary spherocytosis and autoimmune hemolytic anemia. The next one is target cell. So in target cell, the hemoglobin is targeted, that is concentrated only in the periphery and the central region of the cell. So it in short, it appears as a target. So it's mostly seen in sickle cell anemia and thalassemia major. The next one is sickle cell. When the RBC is thin, elongated and slightly curved, then the cell is known as the sickle cell. It is mostly seen in sickle cell disease. Sickling is induced in low oxygen state. The next one is schistocyte or fragmented cells. These are mostly irregular shaped, triangular or helmet shaped. These cells are in the process of fragmentation. It is mostly seen in microangiopathic hemolytic anemia (DIC). The next one is somatocytes. In somatocytes, the red blood cells have slit-like area of central pallor. Normally, we all know that RBC has 1/3 of central pallor, but here it has slit-like area of central pallor instead of 1/3 of central pallor. So it's mostly seen in hereditary somatocytosis and liver diseases. Next one is acanthocytes. When the RBC possess irregular large coarse spicules having sharp ends, is known as the acanthocytes. And these cells are seen in pyruvate kinase deficiency, vitamin E deficiency and liver diseases. The next one is burst cells. When the RBC projections are numerous, uniform in size and regularly spaced, it's burst cells. It is mostly seen in uremia and drug-induced hemolytic anemia. Rollax formation. Red blood cells sometimes align in aggregates resembling stack of coins. It is mostly seen in multiple myeloma, Kala-Azar. In this condition, ESR rate is very high and globulin is also increased. The next one is polychromasia. Polychromasia is term used when the RBC cells stain bluish red with Romanowski stain. So till now we have seen about the various alteration in the size and shape of RBC. Now let's see the various inclusion bodies present inside the red blood cells. The first one is basophilic stippling. When the RBC contains coarsely granular blue-black inclusion inside it, it's known as the basophilic stippling. It's mostly seen in megaloblastic anemia, lead poisoning, thalassemia, and sideroblastic anemias. The next one is Cabot's ring. So when the RBC contains oval, circular, or a figure of 8 shaped ring inside it, it is known as the Cabot's ring. It's mostly seen in megaloblastic anemia. Next one is oval jolly bodies. These are the nuclear remnants of chromatin material. It's mostly seen in megaloblastic anemia or in post-splenectomy. Okay, so that's it for today. Bye guys.